So hi, my name is Vicky Levesque. I am a new scientific researcher in soil health and microbiome at Canville Research and Development Center. So bonjour, mon nom est Vicky Levesque. Je suis une nouvelle chercheuse en santé et le microbiome du sol au Centre de recherche et de développement à Canville. So today I will present you the presentation in English. But if you have any question in French or in English, I will be happy to answer you. So today I would like to show you a quick view of my background and some results of my PhD studies to see what I did. And then I will show you a quick view of my research interests that I would like to do in Nova Scotia. So yeah, I just want to mention you that I grew up in a dairy farm in Quaticook. So Quaticook is located in the south of Quebec, the province of Quebec. And I would like to mention you that I did my university studies at Université Laval. So I am an agronomist in soil science. I did a master's degree in soil and environment. So during my master's studies, I evaluate the potential of different type of constructed wetland to treat greenhouse effluent from tomato crops. And then I did a PhD study in agri-food microbiology, where I evaluated different biochar amendments on the activity and the microbial diversity and on the yield of tomato and sweet pepper. So before to show you some results of my PhD studies, I would like to make sure that you know what is exactly biochar. So biochar is from charcoal, from organic residue, for example, from wood residue that we incorporate in the soil. And according to the literature, we saw that the addition of biochar could improve and help soil health and also the fertility. So why this is the reason we were interested to study biochar. So how we made biochar? So we use different organic residue, for example, wood chip that we incorporate in the pyrolyzer, and we burn the material at high temperature between 400 to 700 degrees Celsius at low oxygen level, and then we produce the biochar. So during my PhD studies, I evaluated 5 different types of biochar, 3 from maple bark, 1 from pine chip, and the last one from willow chip. So you can see the acronym of the 5 different biochar that I used during my PhD, and I will show you some results from these biochar. So the first experiment that I would like to show you that I did is the effect of the biochar and their application rates on the plant productivity. So here I show you the result from a sweet pepper crops. So we can see clearly that the addition of the biochar, when we compare to the control, we increase the shoot biomass of sweet pepper. And yeah, again, according to this picture, we can visualize the effect of the biochar on shoot biomass of sweet pepper compared to the control. Interesting fact is the addition of the biochar in the growing medium also increased the fruit yield of tomato and sweet pepper in our studies. So here I present you when we reduce the fertigation level at 50% of the recommended fertilization level. And we compare the bar chart treatment to the control with the complete fertilization level, we can maintain or increase significantly the fruit yield here of sweet pepper compared to the control on the full full fertilization. So these results are very interesting because they showed that when we when we add biochar in a growing medium, we can increase the fruit yield, especially a sweet pepper, but also we saw some effect on tomato crops. So we can reduce, so we are able to reduce their fertigation level without have a negative effect on fruit yield. So it's a point, it's a very interesting point that we saw. And in addition, we saw in these greenhouse experiments that the addition of the biochar can improve the water and nutrient retention in the growing medium. So this also these results show that we can also reduce the nutrient loss by leaching. And the other research project that I would like to show you is the effect of the biochar and their application rate on the capacity of the mycorrhiza to colonize the leek roots. So the reason why we were interested to evaluate this effect is due because mycorrhiza is a microorganism very important in the soil because they can help plant grow during stress conditions. For example, during a salt stress or drought stress, and also when the nutrients are limited in the soil. So according to our results, we saw that the addition of the biochar in a growing medium had no negative effect on these microorganisms, and also the addition of maple biochar improve the capacity of mycorrhiza to colonize leek roots. So these results that could be interesting to evaluate with other crops to see this combination mycorrhiza with biochar amendment on the roots, on the colonization, on the capacity of the mycorrhiza to colonize the roots of plants. The last project that I would like to show you some result is from a long incubation study where we evaluate the effect of the biochar on the mitigation of CO2 and N2O emission in the fertilized clay soil. So according to the result of this study, we saw at the end of the incubation the biochar amendment reduced significantly the CO2 emission when we compare to the control, and a significant mitigation effect also was observed when we add biochar in a clay soil for N2O emission. So this is very interesting for the environment, but also on the nutrient use by plant. So other research need to do in the field experiment to see their effect on greenhouse gas emission and also on nutrient use by plant in the future project. So, and for my research interest here in Nova Scotia, so is not limited, but so the main objective here in Nova Scotia, I would like to improve the soil health and the fertility of degraded agricultural soil. So the specific objective would be in my research program will be to improve our knowledge on management practices that influence physical chemical properties and microbial diversity as it relates to soil health. Also, I would like to use beneficial microorganisms as allies to reduce abiotic and biotic stresses in crops and to improve the efficiency of the nutrient and water utilization. And then I would like to investigate innovative clean technologies such as biofumigation, soil anaerobic disinfection, and our different organic amendment, for example, compost and our biochar, to optimize the yield and the productivity of the crops. And then I would like to mention you that I am involved in a current project of Keith Fowler. He is the principal investigator on the development of clean technology to mitigate the re-emerging threat of apple replant disease. So the objectives of this study is to replace the agrochemical soil fumigants that have an adverse effect on human health and also on the environment. And the other objective is to improve the soil health here in Nova Scotia. So yeah, it was a quick view of my background and also my research interests. But I will be happy to discuss with you for other opportunity and how could help you in your different research program, and also to improve soil health and fertility here in Nova Scotia. So you have my email address in this slide, so you can reach me anytime if you have any question or any suggestion to collaborate. So I will be enjoyed to collaborate with you. So thank you again for your attention, and have a good day. Bye-bye.